Today he will be presenting his talk titled Shifting Perceptions Around COVID-19, Effects of Vaccination. Thank you, Dr. Ellis.
Thank you, Allie, and thank you for that introduction. And I also want to emphasize that I'm not a medical doctor, I'm not a physician. My doctorate is in microbiology and immunology. So anything that I tell you today that has to do with vaccines, treatments, those types of things are what I've learned throughout my career and what I've learned recently from what has been published about the vaccine that we will be talking about, the vaccines, the viruses. Next slide, please. And I also want to emphasize that there is so much information that we have seen published in the last year regarding the SARS-CoV-2 virus and COVID-19, the disease in humans that this virus causes, that there is no way that we're going to touch on a huge part of that. I'm picking out the parts of the information that is available that will help us understand the current vaccines, the way those vaccines were formulated, the way they were produced, and then how those vaccines are going to help us with conquering this particular disease. And also, I want you to understand that what I tell you today is what I have found to be accurate today. And some of the facts change. So as we go through this in the next weeks and months, we may find more to add to the facts, or some of the facts that we think are absolute truth today may be shaded a little bit and changed a little bit as we go forward and as we gain more knowledge. Don't think that I'm going to tell you things today that are not the truth. I'm sticking with the truth. And just because something may change doesn't mean that I didn't tell you truthfully today what is available. So we're going to help sort a few snowflakes out of this blizzard of information. Next. Definitions that I think will help us understand what we see in the news, what we hear, what we read, and what I'll talk about today. First of all, the virus itself has been named the Severe Acute Respiratory Syndrome Coronavirus 2. That's a long name, a lot of words, and it's been abbreviated to SARS-CoV-2. So SARS, Severe Acute Respiratory Syndrome, CO for corona, V for virus, and 2 because it's the second one of these viruses that have been discovered. And I'll talk a little bit about the first one in a moment. The disease that this virus causes is COVID-19, coronavirus infectious disease 19, because it was discovered in 2019, just shortly over a year ago. December 2019 was when this virus, or when this disease was first identified and named. Next. Then there's also the original SARS virus, same definition, severe acute respiratory syndrome. This was discovered in November 2002 and ran its course in July 2003. So it only lasted for about 9 months. It was a respiratory virus, just like the one that we're talking about today is. And it did cause a little over 8,000 cases and only 774 deaths. When we compare this to COVID-19, we see a huge contrast. And that contrast was in the spread of the disease and also in the severity of the disease. And no one has really defined why that virus died out like it did, but it just ceased to exist pretty much in July 2003. It's also a coronavirus, so it's part of this big family of viruses that structurally are named coronaviruses. And there are hundreds of coronaviruses in humans and in many, many other species of animals. Next. The third virus that I'll mention is MERS, Middle East Respiratory Syndrome. This is another coronavirus that was discovered in 2012 in Saudi Arabia and now is in at least 27 countries. It has not spread nearly as fast as SARS-CoV-2 has spread over the last year. And you can see that it's only in 27 countries. It also has infected hundreds of people and has not caused the amount of deaths that we have seen at all from COVID-19. The animal, it's a virus that is transmitted from animals to humans, and it is transmitted by dromedary camels. So where the camels are, that's where you would expect to find this particular virus, and that is where this virus is found. Next. So a little bit on statistics on COVID-19, and these statistics were what I could find as of yesterday. In the USA, we've had over 24 million cases and now over 400,000 deaths. In Colorado, we have about 5.8 million people total in our state, at least according to the 2020 census, and we've had 380,000 cases of COVID-19. We've had 5,473 deaths, but if you take away some comorbidities, in other words, some underlying illnesses that may have led to the deaths and COVID-19 was just present, the net is 4,579 deaths. That's a lot of deaths in a short period of time in our state. In Larimer County, we get a little bit closer to home, 17,130 cases and 177 deaths. So we're right about 1% as far as the death rate is concerned. Next. One thing that I think is really important that you understand, and I came across this information very, very recently, so it is fairly new information. I don't know, I could not find how broad a spectrum, how many people were surveyed in this particular study, but the averages are really dramatic. So if you're 65 plus and you compare your susceptibility to COVID-19, the disease, with 18 to 29-year-olds, this is some very startling data. If you're 65 to 74, you're 5 times as likely to be hospitalized and 90 times as likely to die from this disease than an 18 to 29-year-old. 75 to 84, the hospitalization goes up to 8 times as likely and 220 times as likely to die. And 85 plus, 13 times as likely to be hospitalized, 630 times as likely to die from the disease. So there is definitely an age associated hospitalization and death rate that increases the older you are. So keep this in mind as we go through this, and we'll have some questions about this probably at the end. Next. So the main thing that we want to focus on today is vaccine development. What happens when a vaccine, any vaccine, is developed? And let's stick mainly to human vaccines. So first of all, if we're going to have a new vaccine developed, go first through laboratory studies and then go on to the animal studies. The animal studies then would see if in fact an animal model can be developed that would exhibit the disease, and then if a vaccine will help to protect the animals from whatever that particular disease is. If the animal studies look promising, then you would go into phase 1 of human clinical trials. A small group, a few hundred people, would be tested with the vaccine. And this is mainly for safety. Is the vaccine safe in healthy individuals? These people are selected because they are healthy to start with. They don't have underlying health issues. So we're starting with a group of people, small group of people that are very healthy. Then if that works out, then you go to phase 2 clinical trials. You would expand the numbers of people. You'd still be definitely looking at safety, but you would also start to look at efficacy of the vaccine. And then the third is where you get into 30 or 40,000 people that are being vaccinated with the vaccine. You would have various age groups. You wouldn't have probably very young children unless this was a childhood vaccination that you were studying. You probably would not have pregnant women in the study, but you would have other healthy individuals. And here, depending on what the vaccine was, but in most cases, you would have a good cross-section of ethnicity, gender, and ages of people. If then everything works out really well at that particular point, safety still a primary factor, then you would seek approval for that vaccine. Once you were given approval by FDA or whoever it is in your particular country that would approve that vaccine, then the company would start to produce the vaccine. They are not going to invest billions of dollars into any vaccine that may not prove out, that may pass phase 1, phase 2, but maybe wouldn't pass phase 3 clinical trials. So they're not, and even if it did pass, for some reason, maybe it wasn't going to be approved. So they're not going to waste their money on that. This would all take 6 to 15 years. 6 is the really low end of the spectrum, and 15 is probably a lot more realistic. Next. So with the SARS-CoV-2 vaccine that is meant to help protect against the disease that we're trying to fight here, they did the laboratory studies and the animal studies. They did phase 1 clinical trials, phase 2 clinical trials, and phase 3 clinical trials. The same numbers of people, the same safety, and the same other aspects that would be necessary to get a vaccine approved. But at the same time, because of backing by our government, President Trump put in the Operation Warp Speed to help vaccine development of this particular vaccine. Then the companies were able to start production of the vaccines that they were in the clinical trials with. So they started the production early on to build up millions of doses, and with the hope that these vaccines would work out. Like I said, safety was very, very high on the list, as it always is. Then the clinical trials with 30 to 40,000 people, cross-section of our population in the US, so that we would know that these vaccines would be safe for most people that would take the vaccine. And then after this all worked out, in only 8 to 9 months after the original studies were started, there was an emergency use authorization, an EUA, that allowed these vaccines to be used in the general population. Next. The types of vaccines that we have seen approved under the emergency use authorization in the US are both mRNA vaccines. The M stands for messenger. And the mRNA, the messenger RNA, instructs the protein factories in our bodies. So in all the cells of our bodies that are going to produce anything, the proteins that they produce are built from amino acids, and the mRNA instructs the ribosomal RNA to tell the ribosomes, which are the actual factories that produce proteins, what to produce. So the mRNA tells the cells in our bodies that we want to produce some of the SARS-CoV-2 spike protein. That the mRNA then sends that signal, and the protein factories start producing the spike protein that is on the outside of the virus itself. This spike protein is what allows the virus to attach to cells in our body, to then be taken into the cell and takes over the cell machinery to produce more virus, if this is in fact the SARS virus itself. But with the vaccine, what it's doing is only producing the spike protein. And those cells are producing that protein and then releasing it into the body. Next. The immune system, our immune system is a very, very unique system in that it recognizes self versus non-self. In other words, our immune system should not be producing an immune response against anything that is self, anything that is identified as part of your self. But anything that's non-self, such as a foreign body or a foreign protein or a virus, a bacteria, any of this, we should produce an immune response against that because it's not part of us. So therefore, the spike protein is recognized as non-self, and then antibodies and cell-mediated immunity are produced, which prevent the virus itself from entering our body cells. Next. So we do have some concerns about this, and if anybody has questions about the mechanisms of the immune system that I just mentioned, we can catch those a little bit later, but I put it very simply, and that is how the immune system works. The basis, the very basis of immune response is recognizing non-self and responding against it to not allow it to invade our body versus not producing an immune response against our own cells. So safety, 2 doses, and efficacy, 2 doses. The efficacy of the 2 mRNA vaccines that have been under emergency use authorization approval in the US have up to 95% efficacy. That means people, those 30 to 40,000 people that were vaccinated, some of them, about half of them or a little more than half, with the actual vaccine, and the other half or so with a placebo, everything in the vaccine except the mRNA. Those people that received the mRNA in the vaccine, in other words, the real vaccine, were protected 95% efficacy against the virus and against the disease. So to explain this a little bit better, how this works is that the first dose primes the immune system. Our immune system looks at that and says, oh, here's something new that I better respond against. It's something that's not part of the body that I'm residing in, that this immune system has developed in. Therefore, I'm going to produce antibodies and cell-mediated immunity. And that immunity starts to become measurable in 10 to 14 days or less. But in order to mount a pretty good primary immune response, then it's about 2 weeks at the longest. The second dose then stimulates the body to mount a greater immune response, an enhanced immune response. So we've already started to produce the cells that are going to produce antibodies. We've produced cells that are going to produce cell-mediated immunity. Those are present, and then the second dose says, okay, let's really get to work here and produce a lot more of this. And then next slide, please. That second dose has a higher level of the immune response. It produces a much higher level of antibodies. It produces it quicker, and it also stimulates a much higher level of cell-mediated immunity. Both of those will last longer than if you just take one dose, only the primary dose. And I had a question just a little bit before this whole thing started, was that these doses, if you get, they're both mRNA vaccines, both the Pfizer and the Moderna. But if you get Moderna vaccine for your primary, you should get Moderna for the secondary, and the same thing with the Pfizer. Whichever one you get first, make sure you get that one second. So far, the duration of immunity has lasted 6 to 8 months. And this has been derived from studies of people who have recovered from the disease, as well as people who have been vaccinated. We don't know what the extent of that immunity is. And it's going to be a while before we know that. So we'll watch this as it goes out over the next months and years to see how long this original vaccination lasts and to see if we'll need boosters from time to time. I'll give you an example of a vaccine that is extremely efficient, and that is hepatitis B vaccine. It's a much different vaccine, much different virus, much different disease. But when that vaccine first came out, everybody knew it was a really, really good vaccine. It had a good, solid protection, and it seemed to last a while. So when it first came out, the recommendation was that you probably would need a booster in 5 years. And then as you went on, it was maybe 10 years and then maybe 15 years. So it just depends on what the vaccine is. Those data were changed for hepatitis B as we proceeded in years on down from when the original vaccinations were conducted. So the same thing will happen with the COVID-19 vaccine. As we get further into this, we'll know more about how long this will last and if and how often we should maybe have boosters with this vaccine. Next. So will this vaccine prevent infection? There is some information that says it may prevent infection, but also you may be infected, but at a very, very low level. In other words, the virus may reside in your body, may replicate to a very, very low extent, and then be gone because your immune system took over and eliminated it before you even knew that the virus was there. But we don't know this again. We don't know how much infection it may prevent or how little it may prevent. We also don't know if the vaccine allows you to, if you are slightly infected, can you still transmit the virus to someone else? These are things that are being studied, that are being watched very carefully, and we will have more information on that given to us as we go forward. Does being vaccinated help prevent the severity of the disease? And this does appear that this really helps. So even though you may have a clinical infection that you could measure, it may not be nearly as severe an infection as if you were not vaccinated at all. And like I said, remember, we're going to get more information on this in the next months as we go forward. Next slide, please. Will either of these two vaccines or any of the vaccines that have been studied or promoted to date and that are understudied to date, will any of these cause COVID-19? Will any of them cause the disease? Absolutely not. In order to have a vaccine cause the disease, you would have to have the virus, a live virus present. And the SARS-CoV-2 is not a live virus in any of the vaccines that are currently being studied. So if you look at a couple of the vaccines, some of them are using a live virus, an adenovirus, which is totally different type of virus. They cause common colds, slight colds. The adenovirus that has been used for a couple of these vaccines isn't even a human adenovirus, causes no disease essentially in people, but it's used as a carrier to get antigens from the COVID-19 or from the SARS-CoV-2 virus into the human, so that people would react to that antigen carried by the other virus. But in no way would any of these vaccines cause COVID-19. There's no way for it to. But what if somebody gets the vaccine and a few days later comes down with COVID-19? Consider the timing. Possibly they were in the early incubation stages of the disease, or possibly they got exposed to SARS-CoV-2 shortly after vaccination before they had a chance to build any immune response against the virus. Some data that I did see, really recent data just within the last few days, showed that in these two vaccines that are out right now, the mRNA vaccines that were being administered in the US, after people got the vaccine and in comparison with people that had placebo, at the same time, when people were exposed in their normal day-to-day activities, the numbers of people that had been vaccinated after about a week or so after vaccination, the numbers of disease really tapered off in that group compared to the placebo or the control group. And their numbers of disease just went right along with what was happening in their general population. So the vaccine really does help to protect us against the virus and helps early on in the vaccination program. Next, please. We've heard recently, if you've been watching the news, that there are some variants of the SARS-CoV-2, and this is not surprising. The SARS viruses, the coronaviruses, will mutate from time to time, and as they are moving from individual to individual, population to population, there are some evolutionary changes that are going to be taking place, and these are called mutations. So in some cases, they can lead to discoverable variants. The way these variants are being discovered mostly is through sequencing of the whole virus and seeing that parts of the virus are different than what the original virus RNA sequence was. But so far, the vaccines are effective against these variants. This is not to say that the vaccines that we currently have are going to be effective against all the variants that may arise in the near future. That's probably not going to be the case. We're probably going to have a few that will not be covered or not be killed by the vaccine. The immune response will not be able to effectively stop those viruses. But the mRNA types of vaccines can be modified to include the proteins, the spike proteins that may be on the variants. So unless there is a mutation that drastically changes the spike protein that then allows it to attach to a human cell and does not allow the immune system to inactivate that new spike protein, our vaccine should still be effective. And this is being watched all the time. I mean, constantly being watched and monitored to see if anything needs to be changed or how the current vaccines are working against the variants. Next. The other thing that people have been talking about a lot lately and a lot in, let's say, the last few months have been what happens when we reach herd immunity. How can we reach herd immunity? And herd immunity happens in people, it happens in animals with various diseases, with vaccinations. It means that you reach a certain level in the population, either through infection or vaccination or combinations of infections and vaccinations, where enough of the population is immune that the virus or bacteria or whatever the disease is can no longer be efficiently transmitted human to human. And it's estimated that somewhere around 70 to 75% of the population would need to be immune to this particular virus before we would start reaching the herd immunity status. That's a long time off. That is months off. And how many months, I'm not even going to guess. It depends on how well people step forward and get vaccinated, how much vaccine can be produced, how it can be distributed efficiently. And the quicker we can get the vaccine out and get the infection rate slowed down, then the quicker we're going to reach herd immunity, and we'll start to see this slow down. Next. The last thing I'm going to say has to do with masks and are masks effective? Absolutely, masks are effective. There's been this is one thing I think that there was a little bit of misstatement on to start with. People at first didn't think they were going to be very effective, and now the data definitely show that masks are very effective if they're properly worn and if they're the proper type of mask. So properly worn means you wear them over your nose and mouth. Guess what? It's firmly established scientifically and any other way you want to look at it. The nose is connected to the respiratory tract. So we're working with a respiratory transmitted disease. You see people wearing masks over their mouth and not over their nose. How are they breathing? Are they only breathing through their mouth through that mask, or are they exhaling a little bit through their nose? Unless they've got their nose clamped shut somehow, definitely they're going to be exhaling through their nose. And if they are infected, they're going to be putting the virus out through their nose. So if when you wear the mask, wear it correctly over your nose and mouth. Adjust it so it stays up. And I've noticed in walking around Fort Collins, different places, shopping from time to time, that, in the grocery stores, it seems to be people my age, 60s on up, maybe 50s on up, that, and I'm 70s up, but these seem to be the people that don't, for some reason, understand their nose needs to be covered. Please, people, cover your nose. The chin is not connected to your respiratory tract. You see people with a big grin on their face and their mask hanging on their chin. That's not doing a thing as far as mask effectiveness is concerned. And some of the worst examples of mask effectiveness, watch NFL football and see how the coaches are wearing their masks. When they need to yell at somebody, the mask is off. But even when they're not yelling at people, a lot of times the mask isn't being worn correctly. So wear it correctly. If you wear a face shield, all that does is just divert the air out around and comes out the side, unless you have a mask underneath it. So again, wear it correctly and wear it properly. Next slide. And that's all. I really appreciate all of you who attended. Thank you very much. And I hope that out of this blizzard of information, there are some snowflakes in there that really have been beneficial to your understanding of this disease and understanding of the vaccines that are available, why we should use those vaccines, why they're safe, and then step forward, get enrolled, be proactive, and get vaccinated as soon as you can. Thank you very much.
Thank you so much, Dr. Ellis. We did get a couple questions. First one is regarding the stats you shared for hospitalization and death. Is that regardless of ethnicity, or is there any sort of change depending on ethnicity that you know of?
So far, what we've seen with ethnicity, certain ethnic groups seem to be more severely affected than other ethnic groups. And what the underlying cause on this is really hasn't been, or causes really have not been well defined. And in this particular study, I did not see where they really defined how broad a group and how much different races and ethnicities were involved in that particular study. But I think the take-home message, look at those numbers. Take-home message is when you get over 65 and over 75 and over 85, your likely severity of disease and likely to be hospitalized really, really jumps higher and higher. So take that for that particular message.
My mute button wouldn't work. Next question is, I've heard that mRNA vaccines have been in development for years. Why haven't we seen them used for other diseases? Do you think we'll start seeing them after COVID-19 for different ones?
Yes, the mRNA vaccines have been in development, even in humans, for at least the last 10 years, and in animals for years before that. So I really fully expect to see more mRNA vaccines under development. One of the possible applications of mRNA vaccines is for anti-tumor vaccines. And since tumors in an individual usually are pretty specific to that particular individual, this would be a great way if the antigenic identities and the parts of the tumor that would help in an immune response could be identified, and then the body could produce antibody and cell-mediated immunity against those tumors. This might be a way to help with that. So this is another possible one. But I think as far as for infectious diseases, yeah, I'm sure we'll see more of these as we go forward. Some, and I don't know if anybody's brought this up in the chat box or not, but there has been a concern voiced by people that, well, once we get this, then this mRNA is going to be in our DNA and it's going to be part of our bodies and then for our bodies for the rest of our lives and possibly passed on to future generations. That is not the case at all. The mRNA will not integrate into DNA in any way. And the reason we may need boosters is because the mRNA will go away after a certain amount of time and then not keep stimulating the immune response by producing the proteins that we need to respond to.
Excellent. Next question is, generally speaking, how long should someone wait to be vaccinated after becoming infected so there is maximum immune response to the vaccine?
I don't have an answer for that, and if I did, I'd want to make sure that it was a very well developed answer that had been published in the literature, and I have not seen that in the literature. Talk to your primary physician and find out from them what they would recommend. Usually, if you're not healthy for whatever reason, when you receive a vaccine, it's not a good time to get that vaccine. So you'd want to be back to your normal healthy self, and then you'd be able to respond better to the vaccine. But talk that over with your primary physician.
Excellent. If there are variants of the virus that aren't covered by the current vaccine, would those individuals who are getting these initial rounds of vaccines need to get a booster?
We'll have to wait and see how that develops. That's a possibility that there may need to be a booster that would include some variant or variants that might be developing. But currently, the primary objective is to get the original SARS-CoV-2 and the variants that are addressed by these vaccines, get that under control. And the quicker that we can get people vaccinated, the better off we're going to get it under control. Then if you start to slow down the virus, the chances of the variants evolving that would not respond to the vaccines are going to be lessened also. So it would serve two purposes. It'll cut down on the numbers of people who are ill and that cut down on the transmission, and it'll also slow down probably the evolution of the virus itself.
Can you discuss the other vaccine candidates and the platforms being used for those? Are they, quote unquote, more traditional than the new mRNA vaccines? And any ideas about how these platforms may influence safety and efficacy?
There are two of the vaccines that are under more traditional types of vaccinology. And so those two would be more like what we're used to as far as vaccines are concerned. There are also two others that I mentioned as I was going through a little bit on the vaccines that are using an adenovirus as a platform. So they're putting proteins from, especially the spike protein, from SARS-CoV-2 into or onto an adenovirus so that that adenovirus will express the antigens from the SARS virus. And then when that is injected, the SARS virus is responded to, as well as some response to the adenovirus itself. So I really don't know where those are in the studies as far as what phase they may be in. Safety, definitely. None of these vaccines are going to be approved if the safety has not been fully established. So that, remember at one of the first, or let's say in the middle of the slides when I was talking about vaccine concerns, safety was first and foremost. So still going through all of the different safety protocols with these vaccines that you would for any vaccine.
Thank you, Dr. Ellis. If anybody has any questions, they can go ahead and unmute and ask those. That would be fine.
I don't have a question, but it sounds like in conclusion, basically we're protecting ourselves for 6 to 8 months in the hope that there's herd immunity, and then it slows down infection. And then it's like a flu shot. If you've started taking flu shots, it seems like you have to take one every year because there's enough change. So the nation going back to normal economically depends on everybody being well, I guess, and dealing with any new variant.
I think you've covered it pretty well. And yes, flu virus, the different strains of the flu virus are very different from coronaviruses. They go through mutations and changes constantly. They're changing all the time, and they do reassortments. So one strain of flu virus will combine with another strain of flu virus and mix and match their nucleic acid material and come up with a whole different strain. So that's why the vaccines that we use for against flu, the seasonal flu, are developed months in advance, but are developed according to what is circulating around the globe and what we think is going to hit our particular part of the world at the time that the vaccines are being made. And that's why every once in a while you guess wrong. Something changes, and the vaccine isn't efficient or as efficient a particular year as it is the next year. So the longevity of the vaccine, this COVID-19 vaccine, is based on what we know to date by how long people have been vaccinated. And we just have to wait and watch and see how long those vaccinations are going to be effective. And we hope they're going to continue to be effective against the spike proteins, that there won't be evolving spike proteins that are totally different. So we'll wait and see, but you summed it up really well.
Dr. Earhart also left a comment in the chat just to say that 6 to 8 months is the longest anyone has seen, and it is possible and likely that it would be effective for longer than that. So fingers crossed on that.
Yes, that's really true, and I hoped I had emphasized that as I went through this, but thank you, Dr. Earhart, for adding to that. That is absolutely where we are right now. We're at the 6 to 8 month time frame, and extending that as we go forward. So it'll be great to see some of the vaccine data that I did see showed a pretty straight line going out. Once the height of the immune response, it stayed up really well 6 to 8 months after the second inoculation. But under normal circumstances, it will taper off some. We just don't know how much. And even if it does taper off, we've still got those memory cells that are part of our immune system that are there, have already been primed, and are ready to respond very quickly if they come in contact with the antigen again.
Thank you so much, Dr. Ellis. Are there any more questions?
Yeah, I have one related to all of that. Is how about those so-called reports of people getting infected twice? Was it really something else, or do we know that they were actually infected twice?
Thanks. That's a really good point. And I have seen data, very, very few data, but where it was pretty well confirmed that a certain individual, and this has been a handful, I haven't seen hundreds or a population or anything like that, where it said that a lot of people were infected, but a person here and a person there seems to have been infected twice. And I did not see any reason, any conclusion, hardly any explanation as to why that happened. But their physicians and the people that were studying that felt that, in fact, those people had been infected twice. I don't know, from an immunologic standpoint, you wouldn't think that would happen, but perhaps that person didn't respond nearly as well as the general population has been responding to an infection. And I think we see that in other populations. Not everybody's immune system is going to respond as well as, there's not a straight line immune response. There are people that respond extremely well, better than the average. There are people that don't hardly respond at all. And those people that don't respond very well to this vaccine may respond very, very well, or don't respond to this infection, may respond extremely well to other infections. It just has to do with how their immune system works. So this is, again, only a little over a year since we first knew about this disease. And I'm sure there's going to be a lot more of this information compiled and put forward so that we can use this to move forward in a more informed manner.
Awesome. Thank you, Dr. Ellis. I'll stay on for a little bit longer, but if that's all the questions we have, thank you everybody for joining us today.